Just to start us off, I mean, I think it's fairly safe to say, not just looking around the specimens in this room, but looking around the people in this room, that colour is something that everyone is interested in, just the colours that people are wearing. So it's something that people find fascinating. In nature, though, colour has different reasons behind it in different types. Could you take us a little bit more through your research into colour in nature and why it arises and how it's produced?
Yes, well, apart from the mechanism for producing the colour, we're also interested in the function of it. What use does it have in nature? Well, it's aimed at the eyes of the animals in its surrounding environment. Sometimes it can be towards the eyes of its own species, such as to attract a mate. Sometimes it's towards predators or competitors, so it can act as a warning colour. We get the whole range of mechanisms for producing the colour in each case. We don't get one mechanism associated with one function. The other interesting thing is to look at the evolution of these colours as well. We look at a range of animals that are closely related, and we see how the colour has been evolving throughout that group. Sometimes, in case of structural colours, which can actually be very important pieces of technology, that's quite useful for the subject of biomimetics because we want to employ some of these things commercially. So we look at a range of species, and we find that sometimes they can be improving in efficiency through the evolution of the group. So it's important to have a good handle on the evolutionary tree there to pick the right species to try to employ commercially.
Okay, so you've got some sort of images of the work that you've been doing. If you could take us through a little bit more about this.
Yes, okay. So as I explained earlier, a structural colour is something like a prism that divides light up into a spectrum. And in fact, Isaac Newton was the first to discover that animals were using this type of mechanism when he looked at the iridescent feathers of peacocks in about 1704. But it's not until today when we've got electron microscopes and we can see precisely the devices behind those colours. It's great working at the museum because we have such a massive collection of species that are still producing their iridescence, because if you put an animal, or a fish for example, with pigment into alcohol to preserve it, the pigment breaks down and you don't keep a record of that. But the structure's preserved, so you still have these bright iridescent effects even in preserved species, specimens such as in museums. Here's an example of one such structure, a diffraction grating. Now, this is the same as the structure you will find on holograms, such as on credit cards, where white light is divided into a spectrum. And this is an electron microscope image of a hair that contains a diffraction grating. And here's the effect that it has. At the top there, you've got these hairs on the antennae that appear a different colour at different angles, just like a hologram. Below, we talked about the subject of function. These are the animals themselves. Now, they have these antennae and hairs held within their shells, and when they get close to each other, the male releases these hairs and displays to the female, who becomes receptive, and they mate. So this iridescence functions for courtship. We've got anti-reflective structures that don't produce any type of colour, but rather reduce the reflection. So the structure on the fly's eye, in this case it was a 45 million year old fly preserved in amber, it has this very precise structure on the surface that if you put it onto a glass window, you would no longer see a reflection of yourself because all the light would pass through. So we've now put that onto solar panels and get a 10% 10% increase in energy capture by reducing the amount of light that's lost through reflection. We've got layers of thin films. Now, this is almost like a stack of soap bubbles or layers of oil on water. You might have noticed a soap bubble reflects different colours in different directions. Well, if you pile them on top of each other, you would get a greater efficiency of reflection, so it would appear brighter. And that's what lots of animals have. They have stacks of these thin films in various architectures. This is actually a crustacean that's only a few millimetres long. They also preserve in fossils because a fossil is a structure itself in any form, whether it's a bone of a dinosaur or the shell of a snail. And the structural colours are structures anyway. So if the preservation is fine enough, you'll also preserve these type of colours. So this is a 50 million year old beetle. We break the rocks open, and some of these look like living beetles. The colour is so dramatic. And we've looked at them in the electron microscopes and found, again, these stacks of thin films. And in fact, the oldest colour that we found dates back to 515 million years ago, where we've managed to accurately colour this ancient scene that would have appeared in the seas at that time. And most interesting at the moment for industry are the photonic crystals. This is the structure that causes the colour of opal. So opal is completely transparent material, but it has this microstructure, a packed array of microspheres that interact with light waves to produce colour, to reflections that we see as colour. Now, if you have a close look at that structure, you'll see it will reappear here in a weevil. So we've got opal, exactly the same structure occurring now in the weevil. And here's a range of species of those type of weevils, again with that same type of opal, but also the inverse structure, which you can see there. And we're getting all sorts of ranges of architectures of photonic crystals that are producing bright iridescent colours in nature. And Francesca is going to cover butterflies in more detail soon. Just as an example, here's the range of architectures that you can find on a butterfly scale, just on a single butterfly scale, invisible to the human eye, which can be broken up further into much, much more detail in the electron microscope. So they produce a whole range of colours and a whole range of effects, and we can look at those structures and see how they change gradually through evolution, to produce different colours, and sometimes can go on to produce different functions.